We are about to start, so if you don't mind to take a seat. First of all, let me welcome you again to the 2025 In Vitro Biology Meeting. My name is Piero. I am the president of the society. I'm happy to see so many familiar faces back. At the same time, I would like to welcome new attendees that they are attending this meeting for the first time. Welcome. So I hope that you all had the opportunity to attend both the plant workshop that we had this morning and then also the panel discussion that we just concluded. A quick reminder for you, this following workshop and the panel discussion that we're going to have at 4 o'clock, and also the first workshop that you had this morning, they have been funded by the NSF, so we would like to thank the NSF. And also they have been coordinated together between the Society for In Vitro Biology and PlantGene. This NSF initiative was actually a 2-year initiative. If you attended the meeting last year, you remember that we had 3 workshops. One that was called Mastering Plant Transformation Vector Design. And then we had 2, actually an additional workshop that was about media formulation for plant tissue culture. And then we had a panel discussion that is a panel discussion like the one that we're going to have today at 4 that was called the Culture Club. The workshop from last year are available on our YouTube channel. And there is a QR code that you see on the screen. And I'm going to encourage all of you, and I know that you all have it, to pick up your phone, scan that barcode if you don't mind. And if you're not already following the YouTube channel of the Society, I will ask you to subscribe to that channel. This is a way for us to really keep sharing with all of you content that we create along the way. And it's also a way for us to really help to increase the visibility of the Society as well. So thank you for doing that. I see a lot of phones that they are coming out. So I do appreciate you subscribing to the channel. This was the panel, the workshop that took place this morning. So like I said, I really hope that you had the opportunity to attend the workshop. What we are going to do now is a sister workshop to the one that we did last year. Last year, it was an intro to media formulation and how really formulation for media evolved within time, according to the specific needs of the culture and also according to the development of the technology. What you're going to see today is 2 talks. And the 2 talks are really going to bring to you the most recent development in the use of design of experiments and also what is the use of machine learning and AI in this space. The first speaker that we're going to have today is Nico. Nico is the head of R&D at 4A Bioscience, and the title of his talk is Frontiers Tools for Plant Culture Media Design. And then the second talk is from Micah. Micah is the research manager at Sierra Gold Nursery. And the title of his talk is Practical Application of Tissue Culture Media Design. The way we design these 2 talks is each talk is going to be around 30-35 minutes. And then we're going to open the floor for Q&A. After the 2 talks, we're going to have a coffee or tea break for half an hour from 3:30 to 4. And then at 4, we're going to reconvene in this room for the sister panel discussion that we originally keep calling the Culture Club. And for this panel discussion, we're going to have John Harbell, Giz Maggie, and Greg Phillips. Unfortunately, Bill Gordon-Camp cannot join us since he's not feeling well. And then Micah and Nico are going to be back on stage to be part of the panel discussion. So this is what we have for this afternoon for you today. Like I said, I really hope that you already enjoy the first 2 workshop and panel discussion that took place today. And now I think without any further ado, we're just going to have Nico to present, but before we do that, I just want to bring to your attention something. If you are in plant tissue culture, definitely the name of Murashige is a name that you are very familiar with. I just want to share with you that we learned that Toshio Murashige passed away last year, and we became aware of it at the end of last year. So I just want to share with you this unfortunate event. And we are planning to have a in vitro in memoria in our journal to celebrate the accomplishment of Toshio Murashige. So without any further ado, I'm going to have now Nico to come on stage, and then we can start this plant workshop. Thank you again for being with us.
Good afternoon, everyone. So I'm Nico. I'm the head of R&D at Foray Bioscience. I'll get a little bit into the company and my background. Before jumping in, just wanted to say thank you to Piero and also Micah and Michelle and the folks at SIVB for organizing this workshop. Let's see. Okay. So name is Nico. There's a phrase in medicine called practitioner researcher, which is something that I've tried to build my career around. So originally, my background's in biological engineering, but as a practitioner researcher, it means essentially you're doing science, but you're also seeing patients. I'm not in medicine. I've basically built my career around the rest of biotechnology and the rest of the bioeconomy. So I've worked in horticulture, agriculture, composting, anaerobic digestion, fermentation, a whole range of areas. And I've tried to, on the practitioner side, help build teams and help it build R&D functions. And on the research side, doing various things across soft matter physics, modeling, systems design, and also applied research in the life sciences. So my work has really traversed the tree of life. So I've worked with plants, fish, bacteria, and fungi. So I feel very much at home at SIVB, seeing all of these areas represented today. And I think if I were to sum it up in one phrase, it would be scaling and manufacturing biology. So I spent the last 10 years working in both indoor agriculture as well as agriculture for the broadacre. So we were doing various sizes and scales of bioproduction facilities and also developing biofertilizers for row crops. So at Foray, I wear many hats. We're combining software, biology, and various other areas to protect and restore natural ecosystems. And we've basically been bringing on more than 20 different species, some of them hard to culture species, into culture systems. So a little bit about Foray. So we have a pretty interesting portfolio of work. So almost all of our work begins with cell line development. And essentially, we do tree, we do non-tree, we've worked in model species, non-model species. This is really the beginning of our various other commercial areas. We've done a lot of work in cultured plant products. Fabricated seed has been another one for us. So this is something we're working on actively right now. You can see this as a photograph of one of our collections of seed that we've produced, and then regeneration and propagation as well. We've recently taken some of the machine learning and AI work that we've been doing to support these areas and been funneling that into a new commercial program that you'll see at the end of the talk. Okay, so why are plant culture systems critical? So essentially, we think that tissue and cell culture are really, really crucial for ensuring resilient and well-adapted plant resources. Here's a few great examples. So Taxol, obviously, the Pacific yew is suffering from being nearly wiped out as a species, and so there's companies like Protalix that are developing Taxol from culture systems here. We have also blight-resistant American chestnut. These have also had a lot of ecological challenges, and we're seeing new resilient crop varieties being developed there. West Coast chestnut is also working on tackling new challenges with grafting, so you can get more rapid bulking. And then finally, there are various folks around the world, including at the Cincinnati Zoo and Botanical Garden, working on conservation and rescue of what they call exceptional species. So what's limiting the field today? So there's over 450,000 land plants today, but a very small fraction of them have been able to be studied in vitro. And so we basically have a really difficult, hard to predict plant-to-plant variability. There are knowledge gaps between species, and a lot of these systems are really hard to optimize. And so today, I want to talk to you about some of the emerging approaches that can change this. So agenda, we're going to talk about why is media design still so critical in plant culture? Secondly, what's holding us back? And third, what are some frontier tools and success stories? So why is media design so critical, right? Like we're 80 to 90 years into in vitro plant culture. What's still important today? Where are the opportunities? So I think I'll begin just by sharing, as we all know, chemistry is the language of plants, right? So plants face some communication challenges that other multicellular organisms do not, right? So they have no nervous system, they have a modular body plan, and actually chemistry is the way that plants have developed as a language to communicate against these challenges. And media chemistry is how we as researchers and practitioners can talk to plants in vitro. So this is an interesting kind of dichotomy that I want to share. Have we solved basal salts, right? So if you look for in vitro culture combined with plant and media, we can see the number of publications over the last 70 years has really been skyrocketing. This is not just linear growth. There's some sort of like acceleration going on here. But at the same time, if you look at when the commercially available basal salts have been developed, 90% of them are actually from before 1996. So what we did to get this graphic, we analyzed around 6 large-scale commercially available basal salt providers. They have essentially hundreds of different recipes available, but most of them essentially are based on around 30 underlying recipes. And what's interesting is that so many labs are moving beyond this and pushing the boundaries. So with Foray or other companies, a lot of folks are essentially homebrewing dozens, if not hundreds, of new recipes. And so what does this mean, right? Have we solved basal salts, or has media design innovation outpaced commercial stock lists, or are we settling with subpar formulation? I don't have the answer to that, but I do know that the impacts of suboptimal media design are essentially lower success rates. We have lower yields and efficiencies. You're left wondering if your outcomes are optimal or not, and then timelines and resource use are long and unpredictable. So what's holding us back here? I want to share 4 areas that we think are making finding the right media really difficult today. So the first one is high dimensionality. So you have a really complex variable space. It's burdensome to investigate. Media components, which are different from ingredients, right? You have like certain ingredients that have many components as part of them. Components can be, even more than 50 at a time in a particular formulation. And also components interact. So those two are tricky. There's also long iteration times, right? Like we're not doing microbial culture where you have essentially, you can get a result in one day, right? We're talking weeks, if not months, a lot of the time for results. And then if you're working across species, especially new species, you have poor translation and knowledge gaps. So what are some things that can address these? So what I want to share first is machine learning assisted design of experiments. So we'll get into a few examples of this, but now that we have greater tools and computational resources available, there are ways that we can apply that to DOE. Various omics, multi-omics, and rapid phenotyping. These are really interesting. I'm going to share an example from our work at 4A on phenotyping. And then automation is big as well. So automation is not just physical, but also digital automation, and those can combine in interesting ways. And finally, a new area that's emerged, I would say, over the last maybe 18 months is this concept of AI co-pilots or AI co-scientists, and we'll share a little bit about that as well. So what I'm going to do next, before jumping into these various frontier tools, is just give a little bit of background on AI and machine learning that will help folks sort of contextualize the conversation to come. So let's kind of define some terms. So when we think about artificial intelligence, what it really means is it's the general field. This is the general field of creating systems that can perform tasks requiring intelligence. It launched actually not so long after in vitro plant culture. So this has been around since the 1950s and has had several waves of sort of boom and bust cycles since then. Machine learning, in contrast, is essentially best described in conjunction with comparing it to mechanistic models. So for plant biologists, mechanistic models could be things like, the Monod equation for doing, nutrient uptake modeling. It could be things like, differential equation for growth dynamics. Like the advent of mathematics and equation modeling, this has been around since the beginning of biology. Machine learning, in contrast, instead of being built on equations and formulas, it's built on entire computer programs. And so this is something that wasn't possible until recently. And when we talk about mechanistic models, we basically are referring to what can be called white box models. So it allows you to see inside of what you're building and understand causation and mechanism. Whereas a lot of the time with machine learning models, they're black box, right? They're basically fitting to the data, they're learning the data, but you don't get to understand always what's driving the patterns that you're seeing. And so that's where gray box models come in. These can be really powerful, is where you're combining your mechanistic white box with your black box or machine learning models into the same system. There are a few types of machine learning. So I want to just highlight a few of them. So supervised learning is essentially one of the original ones. It's basically a good example of this would be predicting outcomes from known media designs. You have a training data set, and you have basically the ability to train your model with labeled examples of, okay, this is the input, this is the output, and the model learns over time. Unsupervised learning, in contrast, you don't have to label your data. It's a way to explore data sets. So for example, maybe you want to find hidden patterns in failed experiments and find why they failed. Unsupervised would be a good approach there. Reinforcement is really interesting. It's essentially allowing the computer to get feedback from experiments as they're running and make adjustments. So you basically have this like reward function that the models are optimizing for, and they give feedback and allow you to try different regimes or protocols as part of this learning environment. And then finally, active learning is where the algorithm gets to choose what examples or what experiments to see next. So I've been thinking about this a lot. This is probably one of my most important slides. So what makes an AI or ML practice? So at the very highest level, you have individuals doing foundational machine learning research. So this would be writing the next version of AlphaFold, which is Google's protein structure prediction system, or segment anything, which is for computer vision. It's essentially developing brand new methods or improving on methods in a way that is getting better performance than what's seen before. We actually don't need to work in this area to apply machine learning and build models as scientists. So the next level down is model building. This is becoming easier and easier and easier today. So let's say you have 200 microscopy images that you want to classify. You can use a library like PlantCV. This is an area where today you can actually take many models, like there are papers that do 8 or 10 different machine learning models and apply them to the same data, either individually or in combination, and it allows you to see which one performs best. And if you structure it in the right way and use the right libraries like Caret in R or scikit-learn in Python, you can actually swap models in and out. So this is an area that I think over the last few years has gotten much, much more powerful and accessible. The third level is AI-powered tools. So you have plugins for benchling or plugins for ImageJ. You have modules on platforms like Benchling that are basically, it's not primarily an AI system that you're using, but the software has AI components. And so this is also an area that I think we're going to be using more and more. And then finally, you have your basic LLM apps like ChatGPT or Claude that essentially allow us to interact and talk to these models at the lowest level. So I want to jump in to the frontier areas now. I'll start with design of experiments. As I alluded to earlier, traditional design of experiments breaks down for slow, kind of high-dimensional experiments. You have here shown a response surface method, and you can just imagine this is like 3 dimensions. What if you have 15 or 50? And so these classical approaches, they work well for a small number of factors, but can fail with more. And so these approaches can be, time intensive and hard to explore. So one solution is go parallel. And so this is an interesting one that for me has been on my mind since working in plant in vitro culture. Coming from microbial culture, as I mentioned before, you can often get essentially, your outcome data in a day, right? Whereas with plant culture, it takes longer. And so one solution that is possible here is to overlap and stagger your experiments. Not only that, though, you can have experiments have crosstalk with each other. So maybe you have like one branch of your experimental line, you're learning something, sort of like an early proxy result, and then you apply that to another experiment. And this is actually implemented in an approach from the social sciences called adaptive experimental design, which is basically when instead of making your design up front and carrying it out, you make your design and then there's a few loops. So you basically learn as you go and you're adapting your experimental design iteratively as the experiment progresses. Another approach to design of experiments is actually to use synthetic data. And I'm gonna give a couple examples here that are slightly far afield from plant culture, but I think we have a lot to learn from them. So the one on the left this is a company called Zordi. They are developing robotic manipulators that can harvest strawberries and other soft fruits. And the challenge that they faced is how do you get these manipulators to learn? How do you give them enough experience, right? Like it could take years. And so what they did, they worked with an organization called, I think Lightroom. I might be getting the name wrong, or I think Light Wheel, yeah. To actually create like a video game for training the software. And so there's this great video where you have the software system that runs the strawberry harvesting, and the machine itself can select paths to grab from in the software, and it can do it at a very, very rapid rate. And then the simulation allows you to see, okay, was the software picked, was it not picked? And so this is like a pretty interesting way of generating synthetic data. I don't know what the equivalent would be for plant culture, but the idea of using synthetic data, this is very prevalent in DOE for real world systems today. The other example of synthetic data that I have is a company called Cradle. So they're actually doing in vitro mammalian culture. So they're working with various mammalian tissues, and they're trying to reversibly cryopreserve them. It's very familiar to us. We're doing some cryobiology work in plants, and you basically have this trade-off of like, how can you design the right cryoprotectant solution? And so what they do is they generate a vast quantity of potential cryoprotectant solutions and cryoprotectant molecules, and they have physics-based ability to simulate how they might perform and how they might behave. So again, I think experiments don't have to be in vitro; they can be in silico. That would be the takeaway here. There's this great paper on kiwi culture that came out, where essentially the authors were combining design of experiments with machine learning. So this is an interesting approach. So they started with regular traditional DOE. They created 33 basal salt treatments. But instead of stopping there, they did 3 machine learning sort of post-analyses of their experiment. So the first thing was they used something called neurofuzzy ML to identify which ions mattered. They then used an artificial neural network combined with genetic algorithms to find optimal combinations of basal salts. And then finally, they created these like interpretable rules showing how each ion affects growth using a tool called fuzzy logic. Okay, so omics. So this is the second of the 4 frontier approaches that I'm going to talk about today. So we're all familiar with epigenetics, genetics, transcriptomics, proteomics, metabolomics. Like these are essentially ways to study collections of molecules within organisms and within cells. Today, there are new approaches that actually expand this and look at system level responses and environmental interaction. So some of these are like just coming out, but essentially you have looking at all molecular interactions, including media. You have approaches that just look at the mineral or ion environment inside and outside of the cell. You have culturomics, you have mechanomics, which looks at mechanical forces and responses, and then spatiomics, which is a big trend in mammalian at the moment, where you're mapping which biological processes occur where. So for us, the opportunity is, a lot of these new approaches necessitate the use of ML, and we can use them to map plant-media interactions. That's sort of like one of the big open problems right now. Some examples. So the top, this is a paper that studied ionomic profiles in protoplasts. And then the bottom, this is maybe more traditional, but essentially it's looking at untargeted metabolomics in hibiscus callus. This is essentially a technique that, because of the complexity of the data generated, you often need to use machine learning downstream as well. So this is a slide I'm really excited to share. So phenotyping is related to, sometimes considered a subset of these multi-omic approaches. The goal of phenotyping in plant culture really is to shorten experimental response times and broaden the data available for decision making. So one of the things we've done recently at Foray is we've been able to collect daily images of our callus cultures and look and ask the question, what is the change in area over time? And so for experiments that you might get growth metrics at day 0 and at day 21, what we wanted to ask is, hey, could you get like an area metric every single day? And what you're seeing here, there's a couple jumps due to weekends, but this is essentially every time there's a star next to one of these bars in the bar chart, it means that there is a statistically significant difference in the growth of that callus compared to the previous day. And so invariably, there are some outliers, but this allows us in different experiments where we want to have a quicker result to look at growth by using area as a proxy. Okay, I want to share a couple examples of physical and digital automation. So when we're doing, in vitro culture, a lot of the time when we hear the word automation, we think of very large liquid handlers. And that is certainly something that exists. You can see some large-scale implementations on the top right image and the image in the middle, where essentially you have the ability to run these pieces of equipment remotely. So Emerald Cloud Lab is a great example of that. CABI out of the University of Illinois is more of a plant science focused example. But you also have tools like Briefly, which is allowing scientists to enter instructions for the various equipment and machines in the lab, and the instructions can be entered in a natural way instead of needing to be written in code. This goes a little bit further. So we're seeing a wave of essentially smaller scale, lower cost, accessible types of robotics and equipment. So on the bottom right, we have GOSH. So GOSH is actually sort of a community biology initiative. It stands for Gathering for Open Source Science Hardware. And their goal is essentially to make open science hardware ubiquitous by sharing designs for affordable and accessible lab equipment. So they've had a number of gatherings. It's essentially community-driven. Some of the things that can emerge from approaches like this, so in the middle is a horticulture seeder. So it's a small seeding line developed by a company called Rooted Robotics. And what's interesting about them is they have used a different approach to material selection, a different approach to design, and they've been able to provide these machines at a lower price point and be able to meet growers where they're at. And so I'm really interested to see if there's examples like that that could be applied for cell culture and tissue culture. What I want to share on the far left, this is a company called K-Scale. Similar to the synthetic data, this is sort of an example from outside of in vitro, but I want to bring it in. So they actually develop like 4 to 5 foot tall robots, and they're for various industrial uses. Again, though, the software part is the hardest element. And it's really expensive to be developing software for these like massive, massive robots. And so what they've done is they have miniaturized their hardware so that it fits on a desk, and any developer can basically access one at a fraction of the price and do development. And so these are some of the things that are going on in physical and digital automation that I think are worth paying attention to, especially around lower cost, and miniaturization. Okay, so now I'm going to talk about AI co-scientists. So we're seeing an explosion of generalist tools that can be used as co-pilots for doing science. I'm sure everyone is using LLMs. You have chat-based LLMs. You have deep research. You also are starting to have generalized scientific research tools like FutureHouse that are trying to help folks answer questions better and do science in a general way for all fields. What's very interesting, though, is that you have a lot of these tool makers that are specializing. So, for example, FutureHouse, they actually released a molecular synthesis AI tool, and it can essentially allow you to design new molecules for new purposes. And so the question is, why would a company that is developing tools for all of science decide to select one domain and build there? A similar example is with Google, right? Google has a lot of different general purpose AI offerings, but they recently developed a program that looks at how apparel drapes, and this is what allows them to have a try-on fashion at home part of their app. And so what's going on here, right? Like, is there essentially a use case for more and more domain-specific knowledge models and constraints to be built? And we think there is. So there's this question, should in vitro plant culture have purpose-built tools too? And I think this is essentially an opportunity that we're really excited about. Oops. So a couple AI co-scientist trends. So we have the rise of a lot of AI lab partners, so moving beyond assistants into co-experimenters. We have self-driven science, where you have autonomous research cycles. You have multi-agent collaboration, so not just one AI, but multiple of them. And then you have dynamic interactivity, where things are being adjusted in real time. I mentioned before some other trends, essentially being able to test multiple ML models at once. And then finally, this one is kind of interesting. You have what I would call inside out, and you have outside in AI tools calling each other. So for example, within ChatGPT, you can now access your Google Drive, and it will learn and get context that way, and that's sort of a outside-in approach. In contrast, you also have benchling. They're an inside-out approach where you're inside benchling, but you can call various tools and various other LMs. So what I want to close with is sort of a little bit of an overview of the AI co-scientists that we've been building at Foray. So this is called Pando. It is named after the giant aspen that has, essentially, it's the largest organism in the world. And that's essentially a way for us to get at this question of more and more unconventional species and being able to bring more biology into culture. And so on the left, this is sort of what we're building in terms of our homepage. What does it do? So we allow scientists to do more and better work in less time, fewer iterations, reduce trial and error, and then ultimately get products to market faster. The areas that we're basically working with here include project creation, research goal setting, library management, protocol building and refinement, troubleshooting, and insights across species. And these are sort of how the areas we've been building at Pando map onto the challenges that I shared earlier. So with high dimensional systems, we have, we're building systems for guided experimental design. Poor translatability and knowledge gaps. This is where a lot of the features focus on, but essentially literature review, community knowledge base, protocol development, and troubleshooting support. So this is a teaser of sort of what Pando looks like today. And I've got a QR code if folks are interested in scanning. I'll just wait for this to run. Okay. So to conclude, AI and ML, they're driving progress across every single one of the 4 areas of frontier tools that I shared, either by powering them directly or interacting with tool inputs and outputs. It's now really become accessible for plant scientists to work in tiers 1 through 3, which, to refresh, are model building, AI-powered tools, and LLM apps. And you can do this without decades of training. I took a class a year ago that basically had us doing this in 12 weeks. We then have unlocking trends. So I'm interested in unlocking, answering more questions around getting more media formulations online and having greater translation to what's available commercially. The new omics approaches, as well as predictably working in new species, both remain a challenge. I think these are areas we want to keep working on. And then finally, AI co-pilots. They offer an opportunity to collaborate with and augment, but not replace, scientists. Okay, and so to close, thank you again. You're going to hear from Micah Stevens next, and he's going to kind of share a little bit more about the space. And this is sort of our summer picnic last year from Foray. So thank you.
Okay, thank you, Nico. Do we have any question in the audience for Nico? If you do, please raise your hand and we can address them now.
Awesome. Great talk. Thanks, Nico. I had a quick question. So I don't know if you saw recently, there was this sort of study that came out where they pulled together all of these world's best mathematicians to try to come up with mathematical problems that would stump these LLMs. And sort of in the beginning, the LLM started asking for more clarification, or like, oh, let me build a simplifying the problem to train myself to answer the real question. So for like Pando and some of these other LLM tools specific for in vitro culture, is there a way to, what am I trying to ask? So is there a way to gauge the confidence of the answer that it's providing based on the information that it has? I mean, is it able to say, hey, maybe I need more information, or I'm only 85% confident on this media formulation that it's predicting? Does that kind of make sense?
Yeah, for sure. I mean, I think like this is a question about prediction in general, right? For all these tools. What we are interested in is getting as quantitative as possible. So it's not just for like, what should my protocol be, but also when you're looking at like asking a question in the literature, where did it actually come from, right? You don't just want like, here's 10 sources and we think that they're saying X. You actually want to say, okay, it's from this location, this location, this location. Predictive tools, in terms of like how you build them, there's various levels. And I think internally, you're always going to be assigning a score, essentially, to that. And it's a matter of like, how do you make those scores meaningful to the user?
Thank you so much. I'm Kelsey, a postdoc from Virginia Tech. I was just curious if it is feeding off of what is published, or more so, can you put your own research into it? And where would the complexity when it comes to different like sucrose providers and batch numbers and having differences when it comes to something like that feed in?
Are you speaking in general terms or for?
Yeah, it just seems like media formulation is so complex, and like if someone on one side of the country or the world used one sucrose provider, you'll have a completely different result if you get it from somewhere else, or even a batch is different and you'll get something completely different. And even if this as saying, oh, yeah, use this media formulation, and you try it, and like, what is it?
Yeah.
I don't know. Like, it seems like such a complex issue that would pull from different sources without having necessarily all of the information, if that
Yeah.
makes sense.
Yeah, that's really interesting. We think a lot about this on the biology side, right? So you have your cell line, but actually that's from a very specific explant, which is from a very specific genotype. And so one of the ideas that we've been talking about is like, how can you sort of track that information so that these systems can learn from it? I think what you just shared on like, hey, you know, this same product from two different companies or two different batches even is like going to be different. That's a really great insight. And I think in principle for folks who are building these types of tools, I would look at it the same way that you look at differences in biology.
I saw
Hey, nice talk. I'm Ken from Oregon State University. So for media optimization, if you're working with like a new species, you can go through the literature and maybe find some related species and then narrow it down pretty close to some recipes that you think might work for your plant that you're trying to grow. And then you can design some multi-factor experiments like DOE that compare like 5 different factors. And then you can analyze how they grow and take data and statistics, qualitative, quantitative statistics, and then analyze those statistics. And so my question is why do we need AI to do that?
My background is in mathematical modeling, right? And I think that AI is just a tool, right? Machine learning is just a tool. You know, a lot of the time, like, I'll give you a great example. We look at problems where, hey, could we try interpolation? Could we try, you know, neural networks? And like, a lot of the times, these different areas can be interchangeable. I think this idea that we have to, you know, that we have to use AI and have to use machine learning, that can lead folks astray some of the time. I think it's more that you want the right tool from the job, and that can come from statistics or mathematical modeling as well.
Thank you.
Hi there, Greg, also from Oregon State University. One challenge we found with in vitro culture and ML approaches is that, like, for instance, we have hyperspectral cameras and they can see all through the visual spectrum. And then when you get to the annotation phase where a human's involved, we're actually pretty bad at picking out individual features on explants, especially if it's reconverted into a sort of false color RGB image. So, you know, are we at a stage where the computer might be able to pick up features that a human can't? Or do you think that all of these really depend on human input for us to look at features? Thanks.
Yeah, that's a great question. I think for things like hyperspectral, I would actually advocate for human-computer teams. I think some of the approaches that folks have had success with are where training is its own thing, but in practice it would be give the image to the computer first. Maybe it'll get 8 out of 10, and on the ones where it has less confidence, it'll say, okay, let me show this to a human now. I think that the challenge though is like, how do you even get to that level? So to get to that level, that is where I would start to look at synthetic data as one potential approach.
Okay, I think we have time for one more question, if we have an additional question in the room.
Hi, Hi, my name is Sandra Romero. I come from Verinomics in Connecticut. And my question is related to the slide that you show, the experiment with the callus. You measure in the different areas. Did you measure something else? And my question, like color, time that the tissue was in media, time to regenerate, shoots. So I wonder if with all the data that we can collect in media, in tissue culture, it's possible to create a predictable tool?
Yeah, I think depending on the application, everything you said would be really interesting. Like we've looked at color, texture, classification as well, depending on what you're working on. So yeah, absolutely. And I think in terms of building prediction tools, that's one of the big use cases of model building. And so, that's an area that I think a lot of folks will be able to get involved in the near future.
Okay, so if we don't have any more questions in the room, I think let's thank again Nico. Oh, there was one? Hold on. There is one. Sorry. Where is it? Oh, there you go. If you don't raise it.
Yeah, it's me, Zarina Joshi from Tribhuvan University, Nepal. So my question goes like that. Is there any automatic protocol that is designed for scale-up of the media that is optimized?
Scalable media development?
Scale up, yeah.
For media scale up?
Yeah, yeah. For the optimized media, is there any protocol, automated protocol designed for scale up of the media for making volume more?
Yeah, I mean, I think what you're talking about is like a lab in the loop approach, right? Where you're trying one media, you're getting a response of, hey, these are the next 5 ones you should try next. You do those, and then the cycle repeats. So from like a design perspective, those are, approaches that can be drawn from. I think scale up of media, what's interesting is, it's easy to make a large quantity of a small number of media types. But
for example, for byproducts formation in large scale, can we go for optimization of media? Can we go for some automated protocol for volume optimization?
Yeah, I think so. I'd love to chat. Maybe we can talk after this.
Oh, okay.
Yeah.
Thank you. Thank you.
Okay, so thank you again, Nico. I think now we're going to have Micah to join the stage, and he's going to share with us a goal and design of experiments for tissue culture media.
Sorry, a little bit shorter here. Well, hello and good afternoon. Thanks, Pierre, for the wonderful introduction. But yeah, like Pierre said, I am Micah Stevens. I am the research manager at Sierra Gold Nurseries. And today I'm just going to present to you all some different ways that we troubleshoot and optimize media at the nursery. So I'm going to dive right in. Cool. All right, so who we are. So Sierra Gold Nurseries was founded in 1951 with the goal to provide orchardists in the western U.S. With a wide variety of top quality fruit and nut trees. And this is just an image on the right of Walter Berg back in the mid 50s at the outset of the nursery. So what we like to do is combine classic nursery methods with modern precision ag techniques to ensure growers are receiving the highest quality trees out there. And then, like I mentioned, we provide trees to folks all over the U.S., but we also have partnerships with university researchers and private biotech companies to develop new genetics. We have roughly 300 employees working to produce about 3 to 4 million trees a year. And this photo on the right is just a photo from our last employee luncheon from all of our production tissue culture lab technicians. Cool. So in order to grow those 3 to 4 million trees, it all starts with our on-site TC lab that will focus on rootstock production, which then supplies the rest of the nursery with the rootstocks it needs to make all those trees. So our commercial TC lab, it started just as a couple hoods, 4 technicians, and a small autoclave in a break room. And so now we're a standalone facility with over 30 plus operators and a support staff of another half dozen or so. So this translates to us acclimatizing, sending out to the greenhouse 30,000 microplants a day, and so roughly 5 million rootstocks a year. Like I mentioned, we produce a wide variety of crops. So this ranges anywhere from almonds, walnuts, pistachios, apples, cherries, pears, and all the stone fruit, as well as a couple other newer crops that we're starting to work on. And so each one of these crops has their own elite rootstock selection, which must be clonally propagated. A lot of times these are hybrid rootstocks, and there's no other way to propagate them except through tissue culture. So they all have their own specific media depending on the stage and the genotype. So all in all, we have 4 to 5 dozen different rootstocks that we're producing each year. And so just a quick overview about where I fit into this picture. I wear a number of different hats in the nursery, but one of my bigger roles running the research lab is to help optimize and design recipes for both our current rootstock selections as well as newer crops that we were trying to develop. And so when I'm thinking about how to optimize or design these new recipes, I like to take a holistic approach and sort of analyze all the factors that can go into a successful micropropagation system. So here are just some different obstacles that we'd like to consider. So proper training technique is huge. It can be incredibly important for successful commercial tissue culture. So oftentimes, output outweighs plant quality, and this can be an issue. For example, if we have all 30 of our technicians cutting plants differently for the induction stage, we can get widely different results for rooting success, which can negatively impact downstream applications for the nursery. Contamination is also significantly impacted, whether it's systemic, environmental, or for the media. We tend to push the plants really hard to meet these production deadlines. So when you push the plants as hard as we do, endogenous bacteria tends to flare up from time to time, especially in woody crops. So sometimes it feels like we're farming the bacteria as much as we are farming these plants in vitro. We've also found that certain crops prefer different temperatures, and so we're also having to manage and think about the temperatures of our grow rooms. For example, apples like it a little bit colder, almond rootstocks like it a little bit warmer, pistachios like it a little bit warmer with more light. So, a lot of those things, get juggled around and come into mind when we're trying to optimize or improve our, media and tissue culture protocols. And then finally, one of the biggest things is these production schedules that we're on are really, mandated by these X-rate minimums, which means the protocol for each one of our genotypes. So for each one of those, 4 or 5 dozen different rootstocks that we're making really needs to be dialed in. So just briefly, what I've illustrated here is just a collection of factors specific to what goes into the medium and what can impact microshoot health and growth. So just to go through some of these, I'm sure you're all very familiar. So basal salts like MS, DKW, woody plant media, all pretty common for the different crops that we're working with, provide nutrition. Then there's the carbohydrates, either in the form of glucose, sucrose, or even sorbitol. Apples, for some reason, really like sorbitol to provide the carbon source. Gelling agents get formed in the media and control the mass movement of all these other components to and from the plant. And then, of course, there's the PGRs, like auxins, cytokinins, and gibberellins to invoke whatever physiological response we're interested in, if it's proliferation, if it's elongation, if it's the rooting stage. And finally, just, things like the vitamins also sort of, will play a role there in aiding growth. But for this talk, I'm just going to focus on these few that I've highlighted here. First, I'm going to go through a project we did with carbohydrates, since they are the energy source for these micropropagated plants in vitro. And what I like to inspect first when I'm troubleshooting media is typically the carbohydrates. And then I will go through a couple case studies on how we approach developing new basal salts and the potential impact on pH of these. Okay, cool. So carbohydrate uptake. When I'm looking at designing or optimizing our current medium, I look at gelling agents, carbohydrates, basal salts, and PGRs in that order. If we're having an issue, rarely do I immediately go to the PGRs. Kind of what I've seen is if these first three aren't dialed in, I found that modifying PGRs can have little or transient effects in our crops. They might do well for a cycle or two, but a month or two later, you really start to lose whatever benefit you had at the beginning, which can be a big problem. So I'm just going to start off with some of the studies we've done with carbohydrates and the approach we've taken there. The two primary carbohydrates that we use are either sucrose or glucose, usually one or the other, occasionally mixed. So when we're doing this carbohydrate uptake study, we started off with just a 50-50 ratio of sucrose and glucose, 15 grams of each, just to have that sort of standard 30 total grams of carbohydrates. And what we did was this temporal sampling of both our of the plants out to day 21. 3 weeks is kind of our typical multiplication cycle. So from here, we would get back just the concentration of glucose, fructose, sucrose, and a couple other minor carbohydrates. And I guess I should also say, so we also sampled and analyzed not just the plants, but also the medium, because the semi-solid, so I was also really interested in what was left behind, not just what was in the plants. Cool, so what we're looking at here is the results of one of the rootstocks, one of the crops that we sampled. This is SG1. It is a peach almond rootstock. It's high vigor, and we already have an established medium for this variety. So we kind of like to use SG1 as a control a lot of times in the nursery, because it's pretty predictable in how it's going to react. So what we're looking at here in these graphs is just a line plot from day 0 out to day 21 on the x-axis. On the top are the sugar concentrations for the microshoots, broken down by fructose, glucose, sucrose, and total sugars. And on the bottom are the semi-solids. And I think one of the things that really stuck out to me when we first got all this data back was that, as you can see, that there was no detectable sucrose in our plants, which was I thought a little bit surprising. But for none of our time points were we able to detect any sucrose. And then on day 15, we kind of saw our biggest jump in growth, which corresponded to a drop in the sucrose medium. And then sucrose completely disappeared in the semi-solid by day 21. And I don't have any photos, but just have to take my word for it. These plants look totally normal. There was no change from the normal media, which is just 30 grams of sucrose. And still, wrap my head around, not sure how to explain yet the decrease in plant sucrose towards the end of the trial period. You can kind of see that between day 15 and day 21, there was a drop-off in all of our carbohydrates here. Other than that, with the decrease in the medium concentration and the total disappearance of sucrose in the semi-solid, this wasn't enough available to maintain this rapid growth phase. So So then the next crop that we investigated here was this walnut rootstock. It's a hybrid rootstock, VX211. It's a little bit slower growing than SG1. But faster than others, so it kind of makes a good middle of the road comparison here. So similar to the SG1 graph, we have the concentration changes for the microshoots on the top plot, and the bottom plot are the changes in the semi-solids. Sorry. So VX211 was efficient at breaking down sucrose into its base components, which, of course, are glucose and fructose, but not as much as SG1, because there's still some present by day 21. There were still some detectable levels of sucrose in the semi-solid compared to what we saw with our peach almond hybrid rootstock, SG1. But the levels of total sugar in the plant stayed stable in the plants up until day 9, which is kind of when we, corresponds to observing rapid increase in growth. So I should also say, when we tested, we incorporated controls which had no plants in them, still, sampling these through these 21-day cycles, and we never, to test the controls with no fructose was never detected, and so if there's no plants in the medium, we never saw any fructose pop up in these carbohydrate cycles, and the sucrose levels did not change either. So this sort of points led me to believe, and still do, that the plants are actively hydrolyzing these sucrose into the base components prior to being taken up by the plants. And then the third and final rootstock that we investigated was this pistachio rootstock, platinum. It's another hybrid rootstock. It's probably one of the slowest growing crops that we work on. The cycle can take up to 4 weeks. It's just, a little bit slower growing. Again, same layout, except with the addition, we sampled out to day 25 here, because it was slower growing, so we wanted to extend that sampling period just to see, what was happening further into its cycle. And kind of what we saw was that it's just not as efficient at sugar hydrolysis, as evidenced by the later detection of fructose. Fructose wasn't detected in the semi-solid until after day 8, and there were still significant amounts of sucrose in the semi-solid all the way out to day 55. But sort of in spite of these plants still grew normally. They didn't, look abnormal compared to what their normal, growth, carbohydrate medium was, which again is, was 30 grams per liter in their typical media. So sort of led me to believe that perhaps pistachios are, potentially hemiautotrophic, or maybe there's just another carbohydrate that we haven't trialed yet that would support faster growth. So here's just, a summary slide, just a side-by-side comparison of all 3 of our crops on day 21 in the microshoots. So we have on the left platinum, our pistachio, SG1, our peach almond hybrid in the middle, and hybrid walnut rootstock on the far right here. And as you can see, regardless of their growth rate potential, and again, this says SG1 grows the fastest, VX211 in the middle, pistachio, platinum is kind of the slowest. Accumulation, there was still no sucrose detected in any of these crops, which again I found surprising. But there was significant difference in their respective abilities to accumulate carbohydrates. VX211 having the most total sugars by the end of day 21, and SG1 had the least. And again, this probably has to do with their potential growth rate and their ability to break down the sucrose and uptake the glucose and fructose. VX211 also had the most fructose, and then should also say that such high levels of fructose in all of these crops, and especially in VX211, was surprising, because when any of these crops are grown on 100% fructose, they just don't proliferate. They don't grow very well, and a lot of them will eventually falter and die. So it's still kind of unclear what role fructose is playing in these plants. I mean, obviously they're getting taken up, but still not sure what it's using the fructose for. So here now we're looking at a side-by-side comparison of all of our crops in the semi-solid on day 21. So just the concentrations at day 21 in the semi-solid. And again, what we found was a strong effect of crop on the carbohydrates that are detectable in the medium, and that sort of matches their respective growth rates. Sucrose was completely used up in the SG1 samples, but is still present in VX211 and platinum. Again, I just think this speaks to trying to find that optimized level and kind of carbohydrate that we're incorporating. SG1 is just very efficient at breaking down this sucrose and taking up glucose, and it's observed in its rapid growth rate. And then also fructose, although, again, this was not added to our semi-solid at all. Remember, it was just 15 grams of sucrose, 15 grams of glucose. It was still detectable in the media, so I think that clearly the sucrose is getting broken down there. So this is strongly suggesting to me that these plants, these microshoots, influence their microenvironment in a way that maybe we don't quite appreciate, and that sucrose is getting broken down, but before it's getting taken up. Okay, so just a quick summary of what we learned from all this carbohydrate temporal analysis that we did. Carbohydrate, vary significantly between genotypes. There was no evidence of sucrose in the microshoot, which suggests it's getting broken down prior to uptake. I think this is further supported by the fructose first being detected in the semi-solid medium, usually around day 3 to 5. And I think this sort of corresponds that when we're working these plants, we sort of see a lag of about 3 to 5 days before we start to see new growth. Again, this is observable new growth. And then once they hit that after that 3 to 5 day lag phase, we start to see some exponential growth before it sort of plateaus off towards the end of its cycle. And I think that the efficiency at which plants can drive hydrolysis of sucrose will sort of determine their growth potential, which for us and these crops peaked between day 15 and day 21. And so I kind of like to think of this as the quicker the plant can modify its environment and have access to its preferred carbohydrates, the better the growth responses will be. And then one final note after saying all this about how it breaks down sucrose, it needs the glucose, it's not using the sucrose, is that growing plants on 100% glucose can significantly change the osmotic potential of the medium. And so when we do that, we have to be cognizant of hyperhydricity in our culture. So just something to be aware of. Okay, so now just to switch gears a little bit to talk about some of the nutrient uptake and basal salt development that we've done. Just going to go through and highlight a couple case studies there. And what I have illustrated here on the right is how I like to conceptualize TC growth, starting with the day they get worked on day 0 and until the end of their cycle. And again, like I just mentioned, we sort of see this lag phase before they really start to ramp up growth into this acquisition phase, and finally in a maintenance phase. So I think in a perfect world, ideally there should be a really smooth transition between these cycles, between subcultures, and minimal disruption to growth. But in practice, that's maybe unrealistic, because inevitably we do see this little bit of a lag before we start to see some active growth. And so also, I kind of think about, these assumptions there's assumptions being that, there's these 3 phases to nutrient uptake in plant tissue culture. During the acquisition phase, it's, can be really influenced by nutrient accessibility. And then if the second one is true, then, minimizing this time here as much as possible will really improve microplant growth. But this growth can often be driven by nutrient availability versus accessibility. Okay, so for this first case study, I'm going to talk about how we set about optimizing a walnut basal salt medium. So walnuts are an extremely high-valued crop, especially in California and throughout the world, but they've been historically difficult to micropropagate on a commercial level. So Driver and Kuniyuki first developed DKW, which has kind of been the gold standard for walnut tissue culture back in the 80s. But as we all know, and as Nico alluded to, you have these really strong genotypic responses that can impact tissue culture success. And with newer walnut genetics coming out, newer hybrids, newer species that are, getting used, they just weren't responding well to DKW. They weren't growing well. And so our goal was to make a new and improved walnut basal salt capable of, micropropagating these newer genetics. Excuse me. So what I have illustrated here on the right is just a graph pulled out of John's paper, where the approach that he was taking was looking at each individual ion and incrementally bumping it up and measuring the response until he felt it hit a level of being optimum. And so we decided to kind of approach it the other way. So we wanted to work backwards, and also wanting to tackle all these nutrients at once. Doing it step by step, one ion at a time, is very time and labor consuming. So we wanted to come up with a way that we can work backwards and sort of back our way into an optimal condition instead of trying to come up to it, while also analyzing all these nutrients at once. So to that end, we wanted to see if simply increasing DKW would work. And the answer to that was yes and no. So here are just 4 photos of what we did originally when we first set out down this course. So on the bottom right here are these walnut hybrid rootstocks growing on 10x DKW. Didn't have a lot of hope, but we wanted to kind of see what that upper limit might be. And as you can see, 10x is definitely too much. Saw very little plant growth, and most of them turned necrotic and died. But then at 5x, we kind of saw some things that were a little bit unexpected. We have some plants dying, yes, but then we also saw an occasional microshoot that would really take off and grow really well. Whereas with 2.5 DKW, we saw really pronounced increased growth. But the downside was that they might have grown better, but they were suffering from quality issues like these small unexpanded leaves and brittle shoots. So after this, we sort of had this aha moment where okay, what does a bespoke walnut nutrient combination look like? So like I said, testing each nutrient one by one is just really time and labor consuming. And to me, I think this misses the interactions between these nutrients. So this is where we set out to sort of develop this better approach. And what we decided to do was similar to what we did with the carbohydrates, where we used this temporal nutrient uptake protocol. There's literature out there. Here's just a couple where other folks have looked at either individual microshoots, walnut leaves, walnut embryos. But when they did this, it was usually just one time point, and it was, in the case of the in vitro shoots, it was at the end of their culture cycle. I think they was at day 34 or something. So to me, I just felt like this missed a lot of the important data points in the middle about when and these nutrients were getting incorporated throughout their cycle. So what we did was we grew our plants on DKW and 2.5 DKW, and we sampled for total nutrients at different time points, again for that 3-week, 21-day cycle. Because what I really wanted to know was if just because the nutrients were there, were the plants using them? Like in the case of the 2.5 DKW, yes, they grew better. They had 2.5 times the DKW. They were there, but were they actually using any of those nutrients? And in this way, it would kind of help us pinpoint which nutrients to target to modify. So here, we're looking at the percent used of phosphorus on the left and nitrogen on the right, broken down by basal salt. So either DKW in red or 2.5 DKW in blue. And what we found here was the DKW was lacking in phosphorus, because if available, they could have taken up more than they could have taken up more. And by day 15, they were using just about all the phosphorus that was presented to them in a normal DKW mix. Nitrogen, however, even at 2.5 times levels, was not taking up any more what was there, so didn't really see a need to modify the nitrogen at all. Cool. Okay, so now these are the percent used for potassium and calcium. Just potassium on the left, calcium on the right. Again, I just sort of shrunk this down to day 15, 18, and 21, but we sampled earlier. I just sort of clipped it to the last 3 sample days. So similar to phosphorus, potassium seemed to be lacking in DKW. By day 15, it already used up 50% of what was available. But calcium seemed mostly sufficient, and probably didn't need to be tweaking. If anything, there was potentially too much, and this can obviously lead to inhibition of other cations. And then finally, the percent used graphs for magnesium and zinc. Magnesium on the left, zinc on the right. And again, what we saw was these microshoots were incorporating and using upwards of 50% more magnesium and zinc by day 15 than what was in DKW compared to 2.5 DKW, which indicated to me that having more magnesium and zinc in the mix would benefit plant growth. I should say we also examined manganese, iron, boron. But apart from manganese, there didn't seem to be any indication that they needed to be modified. So this slide, after digesting all of the percent used data and looking at the concentrations at each day for each nutrient in these tissue samples, we developed this new basal salt called SFN. And use this ion calculator here on the left to help plug in the milligrams per liter, the amounts of what it should be, and have it spit out what the millimolar ion concentrations could be to see how that compares to other basal salts that are commonly used in woody plants. So on right, it's just a comparison between SFN and these other common woody plant salts broken down by ion. And so just have a few highlighted here as ones that I thought, were the ones that needed to be modified and tweaked a little bit to help and improve walnut growth based on these, percent used and, overall concentrations. So what we ended up doing was almost tripling magnesium. Phosphorus was increased, tenfold. Zinc was increased 4-fold, and manganese went up 6-fold. While the ones we identified all were important, I felt like, I thought P was the largest, phosphorus was the largest, rate limiting step here. Okay, so after we did this, we thought we had a great combination here. We wanted to go back and verify that this approach was working. So we repeated our nutrient uptake study, comparing RX1 grown both on SFN and DKW, everything else being held the same. These are the 2 graphs for nitrogen and potassium, and here we're looking at the concentration on the Y and all of our sampling dates across the x-axis there. And so what we saw and what we found was that, yes, absolutely, when available, RX1 is capable of taking up more nitrogen and more potassium. And this uptake is also, hitting that exponential growth curve at an earlier date. So, we're seeing quicker growth, and that was one of the other things that we were really after. So here are the same plots for, concentration of nutrient of SFN versus DKW for calcium, magnesium, and phosphorus. And similar trend here where there was more uptake of calcium, magnesium, and phosphorus, and at an earlier date as well. So this allowed for a shrinking of that lag time after subculturing and led to improved plant performance, without a deduction in plant quality like we saw when we just did 2.5x DKW. We didn't see those, deleterious necrotic spots or small leaves or brittle tissue. We got really sound, high quality walnut shoots. I didn't put it up here, but so same thing with our micronutrients, boron, iron, manganese, and zinc, that when available, these microshoots were able to incorporate more nutrients. So then just briefly, looking at the percent use comparisons between DKW and SFN throughout the course of the culture period. We see the growth typically plateaus in day 18, and all, are consuming all available potassium and only about a third of the nitrogen. So there's still probably too much nitrogen in there, but when you're doing, a lot of these basal salt, custom blends, it's hard to get some other ions in there without incorporating more nitrogen in some capacity, just based on the chemistry of these mineral nutrients. And then, so also similar trends with our calcium, magnesium, and phosphorus, a rapid growth phase that sort of levels off around day 18 as nutrient uptake slows. But even in the case with phosphorus, FSN, as a reminder, had 10 times the amount as DKW, and there's only 20% left after 18 days. So we really felt that using a temporal nutrient analysis, we were able to pinpoint which nutrients seemed lacking and target only those for modification, which really helped to mollify the effects of simply doubling or quadrupling your just already out-of-the-box basal nutrients. So the result was a basal salt combination that was able to maximize nutrient uptake without any sacrifices to microshoot quality. So after we had this SFN developed, we at the nursery were very excited about it, but also we can be a little biased. So we wanted to test it more thoroughly, and so to do that, there was no one better to pass it off to Chuck Leslie. There, thanks, Chuck. And the Walnut Improvement Program at UC Davis, they maintained an extensive in vitro walnut germplasm collection. So they screened 13 diverse walnut genotypes for us. They sort of fell into three groups, either the pure English walnuts, a microcarpa hybrid, or a Heinzii hybrid. They used 36 microshoots, were selected and split between either DKW or SFN. And they were measuring the growth rate by taking height measurements on day 0 and day 21, and they were also recording branching and microshoot condition. So now I'll just go through their results and hope to further convince you of our approach in basal salt development. So the first group of walnuts we're going to look at here are Juglans regia. These are the English or Persian walnuts that are used exclusively for nut production and are notoriously difficult to tissue culture. So on the left here is a cultivar called Durham. Have the day 0 shoots on top, day 21 shoots on bottom, split left to right between DKW on the left and SFN on the right. And you can see from the photos, the plants looking really healthy in both instances, but the SFN shoots are a little bit taller. And then here are just the plots of their mean growth rates. And then yes, so sure enough, there was really strong statistical evidence that SFN led to a increased mean growth rate. Doubling for almost for both of these cultivars, Chandler and Durham. So the second group that they tested were these Juglans microcarpa by Juglans regia. These are hybrid rootstocks that are most exclusively used for as rootstocks in commercial orchards. And these elite selections are propagated exclusively through tissue culture. There's no other way really to get these propagated. And then for these, they used 5 different genotypes, which are listed here on the right side, and then these plots. And then on the left, again, are the photos of day 0 and day 21, split left to right with DKW and SFN. And although there was no, statistical evidence, statistical significance that SFN led to a, increased in mean growth rate, in all instances, it did have a higher overall mean growth rate. And then finally, the third and final group that they looked at were Jugelin's Heinzii by Jugelin's regia, or paradox walnuts. And again, these are used as rootstocks. Paradox walnut was first developed by Luther Burbank, and the paradox walnut was what John Driver used to develop DKW. And here they tested two different genotypes from this group, Grizzly and VX211. So these photos are of Grizzly, and when we set about designing SFN, Grizzly was the reason why. Grizzly is, an up-and-coming, highly sought-after rootstock, but it was just not performing well at a commercial level on DKW. So this was sort of the impetus for, designing this new medium. So you can see the shoots growing on SFN, almost reaching the top of these containers by day 21, and on DKW, kind of barely coming to the top of the test tube lid. So again, SFN significantly improved the growth rate compared to DKW. Excuse me. And in the case of VX211, the mean growth rate almost doubled. So this is huge when it comes to commercial lab production for increasing efficiency of being able to clonally propagate these, elite rootstocks. So in the end, yes, we felt like this was better. So here's the combined data across all genotypes, and so I can, kind of confidently say that our approach to improve these basal salts for walnuts worked. Culture on SFN medium resulted in greater shoot growth than culture on DKW. SFN had greater variability than DKW, but at worst, it is at least the same as DKW. The groups, the English group and the the Heinzii by English genotypes exhibited significantly better growth on SFN, whereas the Microcarpa by Regia hybrids was similar, but the shoot quality tended to be better on SFN. All right, so that was our experience with walnuts. We were really excited, it worked, and so we're like, okay, cool, let's tackle our next project here, which is citrus. Citrus is a little bit newer crop for us in the tissue culture lab. And when we started working on it, we were getting in all these different hybrid rootstocks to be able to clonally propagate, and quickly noticed very significant genotypic differences in growth and performance. So what I have just illustrated here are 3 of these genotypes, US 812 on the far left, Rubido in the middle, and Swingle on the far right. You can see 812, barely proliferating at all. Rubido having very small kind of stunted leaves, some chlorosis mixed in there, and Swingle looking okay. So we took the same approach with walnuts, but spoiler alert, it did not go to plan. But I'd still like to briefly share what we found and kind of discuss what I think might be going on. I think that's kind of important to discuss. But then here, one other thing we did differently was we incorporated some automated high-throughput phenotyping this time around too. And so in order to do that, too far. Yeah. We partnered with Chimeral to use their chem cradles, and Vladimir's here. They've just been incredible to work with. We've been able to collect real-time growth data during these media trials, as well as more qualitative data like chlorosis and necrosis. And the way that these work is we have our tissue culture vessels here in the photo on the left in the foreground, and they just fit right into these cradles. And there's a camera on top that will read a barcode that links the cradle to that container. And then there's another camera on the inside that is, recording plant pixels, and we just leave these in the cradle through the duration of their growth cycle. And they are, taking photos and recording these plant pixels at, kind of whatever interval we, thought would be useful. In this case, we were doing it once a day. So So we started off with Swingle. It was one of our better performing ones. We wanted to kind of, zero in these CRIM cradles, see how they were working, because it was more, one of our more well-behaved genotypes. So what we're looking at here on the left is the growth curve that we got from our chem cradle data. And I kind of alluded to it earlier with the lag phase and kind of what we're observing, and with the, our naked eye, but with this, chem cradles, we're able to see that, yeah, the plants are growing. It's just really slowly up until, later in their cycle when, that exponential growth rate really starts to kick in. Especially around day 23, kind of before plateauing. Here was Swingle, and these photos on the right are, just an RBG image of the Swingle inside the cradle, and then just a verification of that only the plant pixels are getting recorded here. So after the early success with Swingle and validating these chem cradles, we moved on to another one of our genotypes, rubido, that I kind of thought would be a better candidate for basal salt development trials. We repeated this temporal analysis using DKW and 2.5 DKW as before as our baseline, but then I was very surprised to see that the plants grew much worse on 2.5 DKW. I mean, they were growing okay on DKW, but we were clearly seeing signs of nutrient, deficiencies in these plants. We saw that, chlorosis earlier. And you can see from these, photos, the DKW plants looking good by day 20, looking okay by day 24, but, the 2.5 DKW photos, by day 24, the plants hardly grew at all, and that matches the data we got from the chem cradles and their, their relative growth rate over the course of that culture period. But again, when we looked at the nutrient data, we saw the same thing. It was plants on DKW had lower overall concentrations of each nutrient. I mean, they weren't growing, so not much of a surprise there that they didn't have, much nutrients in them. Here are the concentration graphs for nitrogen on the left and phosphorus on the right over the course of their culture period. And so this led me to believe that nutrient availability is not really what was causing the poor growth in rubido and potentially our other citrus genotypes that were not, proliferating very well. So I wanted to also look at the percent used data, because this is kind of what helped us predict and design what an optimum level of each one of these nutrients might be. So here are, the data for nitrogen, potassium, sorry, I lied. There it is, sorry. Calcium, magnesium, phosphorus, and again, these plants on 2.5 DKW were, using less than 5% of what was available. This just seemed really surprising to me because the plants growing on DKW were, again, showing signs of nutrient deficiency, so you would think by adding more nutrients, not even that much, 2.5 times as much, they would have some better growth. So I had anticipated that increasing nutrients would improve plant growth, or at least not have such a negative impact as it did. But not to be deterred, we designed a couple basal salts here listed as CIT1 and CIT2 in the same fashion as SFN, using this percent nutrient data as a guidepost to help predict what might be lacking. And yes, you can see from our results things that did not go as planned. Both media did poorly, so poorly that they didn't stay alive long enough for us to do even any nutrient sampling. You can see from the graph they grew for about 5 days before crashing, and then by day 22, you can see in the photos there were just not many left. On the highlighted plant pixels on the bottom, you can see that it's just kind of picking out live tissue, and there's not much left. So clearly something else was at play there. So my best hypothesis that these microsuits, again, are changing their pH and their microenvironments in a way that are negatively impacting their ability to access these key nutrients. And I'm really excited about this idea. Through our, both our carbohydrate testing and our nutrient testing, we recorded pH over the medium, or pH of the medium, I should say, but only saw minimal fluctuations in pH between sample days.
However, we had to homogenize the medium, and by doing so, I suspect we masked any changes that might have been happening in the closest proximity to the plants that might be interfering with nutrient availability. So we devised this contraption where I got this really fine point pH probe and suspended it at multiple points above the microplant, around the microplant, and there's a pH logger attached to it throughout the course of its 21-day cycle period. We just started this experiment, but I'm really looking forward to the results, and I think it's going to lead to helping us answer those questions about what it is about these citrus not being able to access some of these nutrients in the medium. Okay, cool. And so just to wrap up, when you're at a roadblock with a recalcitrant species, I'd like to think take a look at the carbohydrates first, as long as the gelling agents. We found significant genotype effects on how carbohydrates are broken down in the medium prior to uptake and in what form they are found in the plant. Again, we never detected any sucrose in the plant. And if that doesn't ameliorate the issue, consider a temporal nutrient analysis. It really allowed us to understand what was missing without having to test each ion one by one. But again, as we saw with the citrus, this is not foolproof. You kind of need a decent amount of material already established, and as with the case with the citrus, availability does not mean accessibility. I suspect with the citrus, there's something with the pH that is locking up nutrients. As with the carbohydrates, we already know these plants can modify their microenvironments. And finally, incorporating an automated phenotyping system has really allowed us to be more flexible and effective with our media trials. And then just one last thing is that SFN is actually available through Phytotech. So if anyone is interested in trialing it, I would definitely be very interested to know what crops you're using and how it worked. I would be very interested in that. And with that, I would like to thank Piero and the rest of the SIVB for the invitation to speak today. Thank you all for being here. And of course, thank you to my lab and collaborators, and I'll be happy to answer any questions.
Well, thank you, Maka. Do we have any question in the room? If so, please raise your hand. I see one. I'm going to have, at the end of the day, a lot of steps. So yeah, one in the back, and then I'll be back.
Thank you. My name is Bakala. I'm from Israel, Robot Institute of Weizmann Institute of Science. Okay. So my question is, as we know, when you prepare media, first we calibrate the pH, and then autoclaving, there is a change in the pH maybe. But this is not common also. I don't know, I have no idea how we can measure, and we didn't measure, but there might be significant difference in the pH after autoclaving. And the pH is also important for nutrient uptake. So is there any ways that you have shown or you have detected a change in pH, and what could be the range? Because the nutrient availability pH range itself in soil is known that is very narrow, from 5.5 to maybe 6 something. So do you have any idea? Because from your graphs in nitrogen and phosphorus, if I am not mistaken, some of the reduction in phosphorus uptake might be due to a big change in the pH in the media, my assumption. Thank you.
Absolutely. Yes. So I think that's why I am so excited about this pH experiment that we're just starting to run. Yes, the autoclaving can change the media. We usually start off prior to autoclaving everything at 5.5, 5.6. After autoclaving, it'll drop down to 5.2, 5.1. So we're very interested in measuring the pH specifically around the microshoot in the medium, because like I said, when we took the pH, when we were sampling carbohydrates, when we were sampling nutrition, we didn't have this really small pH probe, so I was having to homogenize the whole container together, and I think this was masking a lot of what was happening in the immediate vicinity of the microshoot. And so yes, I think you're right. I think these changes in the pH are affecting nutrient uptake, and I'm interested to know in what capacity. And I think having the ability to measure the pH directly on top of the microchute, 2 millimeters away, 5 millimeters away, 6 millimeters away, and kind of getting that spatial reading of how far away these plants are able to impact changes in pH in the media is going to be interesting. Great question.
So mine might be really naive, but since you observed that your citrus plants grew worse on the 2.5x DKW than the 1x DKW, I know that you said you already were observing some sign of nutrient deficiency in there, but what, if anything special, stopped you from trying like a lower level, like a half X DKW? And is there anything to that?
No, that's all. Yeah, great question. And so far, nothing. I guess that was just my own recency bias with SFN and thinking that we needed to go up. It's very possible that we need to go down. All of this citrus work we've just been working on in the past 6-7 months, so this is still work in progress. So yes, it could be that we need to go down. I guess I don't necessarily think of DKW as a high salt medium. So going up, to me, didn't think it was going to be that drastic of effect on plant growth. But yes, I mean, you're absolutely right. There's no reason not to go down to 0.25 DKW, 0.5 DKW, and repeat these same nutrient profiles to see what's happening at the lower levels. No, great question.
Thank you for such a nice presentation and all the details, very meaningful. The question I have is about when you were making a presentation, early slides showed CO2 in the environmental conditions. So I wonder if you are checking or using carbon dioxide levels in your chamber to test these parameters for plant growth. And if you are doing it, can you share some details?
That is a great question. We are not doing it. I know of some other commercial TC labs that like to enrich CO2 in their growth rooms. This is not something that we've explored, mainly for safety considerations. We don't have an area where we can sort of elevate CO2 levels in our grow rooms and have it be done in a way that we think is going to be effective in the sense that it's actually getting CO2 into the plants. I think what might be real interesting is something for like pistachio, which seems to me is potentially hemiautotrophic in tissue culture, would be really interesting to investigate. I will say my one experience with CO2 enrichment was with pear rootstock using a temporary immersion system, where every time it was aerated, it had a regulator on it that was also pulling CO2. And yes, I saw dramatic differences in the growth of these plants when CO2 is enriched in a TIS system. I don't know that it would be the same in a semi-solid system. Yes, and I've done some other harebrained ideas to try to increase CO2 directly in the vessels, which I'm happy to share with you later, but it's nothing that we've done on a grow room level to increase CO2. We just know that it's an important factor, but nothing that we've regularly investigate.
Okay, let's thank again the speakers. I know there are more questions, but we have a I'm trying to wrap up this. We have a coffee break outside, and then we have a panel discussion. So if you come back at 4, we're going to have the panel discussion so we can keep having the question that you had right now addressed during the panel discussion. The coffee break should be out. Let's thank again Mike and Nico.